My talk was one of three of a panel of investigators who have a specific interest in looking at the basic science behind breast cancer disparities, specifically in populations of Hispanic, Latino, and African Americans. My lab is actually focusing on how genetic ancestry has impacted the biology of tumors. So breast cancer isn't just one disease, there are multiple types. And for whatever reason that we are trying to identify, African American women tend to get one of the most aggressive forms. And one of the disparities that we see is in mortality. African Americans die more frequently from this particular type of cancer. It's triple-negative breast cancer. And so what we're trying to do in the unique opportunity that I have at Henry Ford working with Dr. Newman is that Dr. Newman has gone into Africa, various countries, particularly in Ghana primarily, and she has a unique cohort of patients and controls from Africa. And so what we're able to do is assess genetic risk within African populations that can inform us about some unique features in the genomes of African Americans here in the country. There have been many studies that looked primarily in groups of African Americans and identified genetic markers. One sort of characteristic that I see with some of our genomic studies is that genes that are expressed differently in African Americans compared to white women who get breast cancer are in immune-related pathways, meaning they're genes that regulate the immune system. They drive whether or not certain types of white blood cells get involved with the tumor. And what we have discovered is that there is an ancient mutation that actually traverses the entire African diaspora because it's fixed in sub-Saharan Africans in a gene that regulates inflammatory pathways. And so what we've started to do is correlate whether or not this mutation is driving the expression of the gene in tumors. And then when the gene is expressed in tumors, we see a unique immune response. And so what this is allowing us to do is characterize not only this subtype of breast cancer, but also the way the tumor progresses uniquely in women who are of a specific ancestry. We do know enough about immunology in tumors to be able to predict what this immune response is doing. So there are tumor immunologists who sort of very distinctly define for us what to expect a tumor to do if certain types of immune cells have infiltrated the tumor. And so what we're going to be able to do is identify those tumors without actually knowing primarily directly which immune cells are infiltrating. So for instance, if the tumor is positive for our gene marker, then we can anticipate a certain subset of immune cells to be present in the tumors. What that will do for treatment is identify whether or not patients are suitable for immune therapies and what types of immune therapies. So we'll be able to assess whether or not the patient has a good immune response happening at the moment, and so their immune system is actually helping fight the tumor, or if their immune response is helping feed the tumor. So we would be able to identify that sort of dichotomy in patients with our marker. I always underestimate the time, so I'll say probably this time next year we will have characterized the tumor type, and we'll start doing some clinical association trials, with just using the marker to identify patients who would be suitable for specific types of trials. So we don't necessarily develop drugs, but there are enough people who do. And I think that what we'll be able to do is partner with people to identify what population of breast cancer patients would benefit from their trials. We see corollaries of the tumor types that are in Africa with the types of tumors that African Americans have. And so with Dr. Newman's cohort, we will be able to investigate the specific genetic driver in the African populations to allow us to identify those same drivers in African Americans. They've been really evasive to find with the genetic admixture of African Americans with European genetics here in the U.S. And I think that we have this great opportunity right now to sort of bolster the field and make some tremendous contributions. So we're excited.